Hi everybody, it's Dr. Brazier here, and I'm going to go over your first virtual lab of the semester. And what we will be talking about is the Hardy-Weinburg principle. Now this is Lab 1, Population Genetics, and this is going to introduce the idea of evolution. Evolution is nothing more or less than looking at the heritable changes in a population that occur over several generations. As science began to study evolution, they also looked for ways to measure it. How could you tell if a population was evolving or its genes were constant, i.e. not evolving? Was there a way to compare rates of genetic evolution? It took a collaboration between two diverse fields to answer these questions. G.H. Hardy, an English mathematician, and Wilhelm Weinberg, a German physician, independently devised two equations to determine if a population is evolving or if it's stable. As a reminder, a population is all individuals of the same species in a common area. And these individuals share a gene pool. A gene pool is just all the genes in the population at any given time. If there's no evolution, that gene pool will remain constant. And there will be no change in the frequency of the various alleles or how many alleles you have there. The dominant and recessive forms of the genes are called alleles in a gene pool. So let's just kind of review what an allele is. So if you remember back to general biology for number 1 for majors or non-majors, you'll remember looking at basically genetics and looking at offspring and how you would calculate offspring. And in order to do this, you had to understand two concepts. You had to understand what a genotype was, you had to understand what a phenotype was, and then you had to be able to write them out and then determine how they correlated to each other. And so if we're looking at something like rabbits, and then we're looking at, do the rabbits have brown fur? Or do they have black fur? Determining what they looked like was a phenotype. But then, if we wanted to see what was the actual genetics that led to this phenotype, that was the genotype. You can think genetics here, physical characteristics here. Now, you wouldn't necessarily know this automatically. If you did a bunch of experiments, you could figure this out. But we're going to say that the dominant fur, the brown fur, is the dominant phenotype, and the black fur is recessive. And what do we mean by this? Well, it's not really the dominant or recessive phenotypes. It's more attributed to what is known as a genotype. So when we look at this, you are going to get a gene from your dad and a gene from your mom. The genes that you get are actually for that particular phenotype, or the type of gene that you get is called an allele. So in this case, the alleles for fur can be either a big B or a little b. The big is, when we're talking about, means dominant. The little means recessive. A dominant allele means that if this guy is present, you will see it. So this is like a group project. If you have a group project, a dominant person is the one that's loud, that gets heard, and people notice them. So if that person is there, they are seen. A recessive means it's only seen when there are others like it. So for instance, if you're in a group with a dominant person, you're probably not going to be heard if you're a recessive person because you're going to be quiet and that dominant person is going to be talking. The only time you're heard is if there's a lot of quiet people. Then you get to come through. And these are alleles. These alleles are very specific forms of a gene, and they can be dominant and they can be recessive. Now up here I said we had phenotypes, and the brown fur phenotype was dominant and the black fur phenotype was recessive. And what I really meant to say is that when you look at these, you have a big B, and a big B is the genotype. for one type of phenotype, and then you have a big B, little b, and then you have a little b, little b. And so this is what's known as a homozygous dominant, which means it has two big B's, and this one is going to be brown because that's the dominant phenotype. This has a big B and a little b, so this means there's a combination of alleles there, so it's heterozygous. This again will be brown. And then this is homozygous recessive. This is going to be black. So when we use Hardy-Weinburg here in a few minutes when I teach you about population genetics, we are using and looking at what is the frequency of these alleles, i.e., how many big B's are there in the whole population? How many little b's are there in the whole population? And then we can go and say, okay, well, how many big B's do we have in the whole population? How many big B little b's and how many little b's? And then from there, we can compare one generation to the next to see how these alleles are behaving. Are they changing? If the alleles, or how many big B's are there, how many little b's are there, remain constant, then Hardy-Weinburg is in effect because there's no evolution. If you start seeing alleles changing, then you know Hardy-Weinburg is no longer in effect because it is changing. Now, when we talk about Hardy-Weinburg, Hardy-Weinburg can only remain true if 5 conditions are met. First, there has to be no natural selection. What this means is there can't be any environmental pressures that affect survival rates. So a perfect example of this is if you have black mice and white mice, and they're running around a mountainside. They both equally can be seen by predators in a normal environment. But then say maybe we're on Hawaii, and all of a sudden you have a volcano erupt, and the lava comes out, and essentially it dries. And then that background now is black. If you have a black mouse running on it, it's not going to be seen. It's going to be well hidden. The white mice are going to be easily seen by predators. So now the white mice are going to be eaten. This would be an example of natural selection, that the environment essentially is allowing one to survive better than the other. So these are environmental pressures. But we can't have that in a Hardy-Weinburg equilibrium. Remember, equilibrium means no change. We can't have gene flow. What does this mean? We can't have individuals coming into the population and leaving the population. The population has to be kind of removed from everybody and everything. So you can't have things coming freely in and out. It has to be kind of separated. There can't be random changes, which those random changes are called mutations. We can't have those. Mutations lead to DNA changes, which lead to the DNA of the population changing. So we can't have this. No genetic drift. Sorry. No change in the gene pool due to chance. This just means things are kind of moving and changing willy-nilly, just kind of by chance. And that cannot happen at all. And the last one, there has to be random mating, which means people, organisms, whatever, are just mating free with anybody. This isn't like the 70s free love. It just means that the individual in the population are able to choose who they mate with. There's not certain conditions based on that. So if these conditions are not met, the population would not be in Hardy-Weinburg. It means they won't be in equilibrium. Thus, the gene frequencies, P and Q, and the phenotypic frequencies, big B, or P squared, 2PQ, which I'm going to talk about here in a second, that will be changing. So I talked about essentially that this was created by two individuals. And what they did was Hardy and Weinberg proposed the Hardy-Weinburg Equation to calculate the allele frequencies. There are two of these equations. So this first one is the frequency of the alleles in the gene pool. So this is just saying the P plus the Q equal 1. So P is the frequency of the dominant form of the allele. So how many P's are there? What's the percentages of P's in the whole population, right? And P's are, if we were using our B's for fur, would be the big B. How many big B's are there in the population? But we said frequency, so it's kind of like a percentage, or the one here is going to be equal to 100%, but we're doing it in decimal form, right? So basically, however number we have of big B's, or this P plus Q's, which are the little b, equal 1. So these two have to add together to equal 1. So this is just saying how many of this gene do we have there and how many of this gene do we have in the full generation. Now, if you remember, we can have three forms of it here, right? We can have big B, big B, big B, little b, little b, little b. So these, we're going to use the next equation to look at the frequencies of those. So then we can go here, the frequency of the individuals in the population. So here we can look at this and say, well, how many? We have P squared. So if we have two P's, this would be equivalent to big B, big B. Q squared, well, we know the Q would be the little b, right? So little b, little b. And then this guy right here has both of them. So this would be big B, little b. So all three of these would come together to equal 1, or 100%. And so you can find out the percentages just by knowing a little bit of information. For instance, if I tell you what the frequency of this is, well, if it says, 30% of the population is homozygous dominant, well, then PQ, or P square, I keep saying PQ, I'm sorry, P square is 30%, or P is 30.3. And then you can get the square root of 30 to equal P, and then you can find from there Q by adding this into the P plus Q equals 1, and then you add both these into each one of these, and you can find all the information out. So it's basically just plug and play. So let's go through these pre-lab questions. I'm going to go over a few of them, and then some of them you're going to answer yourself. So number 1, what is the definition of evolution at the genetic level? And basically this definition, i.e., is microevolution. And so the next thing is what is microevolution? And so microevolution is essentially change below the level of species. And what does this really mean? Well, it means that microevolution is the change in allele frequency that occurs over time within a population. Now, this kind of goes over what we just went over. In the equation P plus Q equals 1, what does P represent? P is the dominant allele, right? So you could do an example. This would be our big B, right? And what does Q mean? It's the recessive allele. So this would be the little b. If P equals 0.5, or 50% of the genes in the gene pool are dominant, what is Q? What does this mean? And so we can use the whole P plus Q equals 1 to do this, right? I always write these two equations at the top of mine so that I can just look and always refer to it. So P equals 0.5. So then we just add this 0.5 into the equation, right? So if we have 0.5 plus Q equals 1, then what we would do is we would minus 0.5 off of both sides. And then what you would get is Q equals 1 minus 0.5. Oops. Which means Q would end up equaling 0.5. This means that P and Q are the same frequency within the gene pool. They're both 50% of the gene pool. In the equation P squared plus 2PQ plus Q squared equals 1, what does each of the factors represent? Well, if you remember, you could look and just write big B, big B, big B, little b, little b, little b. And then what does this mean? This means homozygous dominant. This means heterozygous. And this means homozygous recessive. What does it mean to say that a population is in Hardy-Weinburg equilibrium? This means population gene frequency is stable, i.e., unchanged, no evolution. So I want you to answer the following questions. What conditions would disrupt Hardy-Weinburg? So what you need to think here is, what conditions do we need to meet Hardy-Weinburg? And so if they weren't met, they would disrupt it. Then number 8, how does natural selection affect Hardy-Weinburg equilibrium over several generations? Clearly state your hypothesis. And then your last question for pre-lab questions are, state what you would predict if your hypothesis is true about the Hardy-Weinburg equilibrium of the population of rabbits after 7 generations, where little f, or recessive bunnies, homozygous recessive, are selected against, which means they die. So what would happen over time? So we can practice it a little bit here. I wanted to give you all a chance for me to work over a problem so that you would be prepared. This is going to be a little bit different than your lab, because in your lab, you're going to get all the potential number of organisms that are there. But in a problem that could be on your test, you would only get the frequency of, say, something like this, which is homozygous recessive genotype. But you can find out all the rest of the information that you need just from that. And so on here, it says you have a sample, you have sampled a population in which you know that the percentage of the homozygous recessive genotype recessive A is 36%. Using the 36%, calculate the following. So very first thing I'd do here, if you all remember, is I'd write my two equations. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. I write that at the beginning, so then I don't have to worry about forgetting them, right? So the frequency of the little a genotype. Well, they've already told you that it is 36%. So it's given. So the little a genotype is 36%. Now, we can write this in decimal form, which would be 0.36, which will help for the second question, which says the frequency of the A allele. Well, here we have two A's, so how do we get the two A's? Well, if you remember the AA, we need to look at these equations and say which one do we recognize this as? And this is the same as the Q squared because it's the recessive, homozygous recessive, right? So here you would do Q squared equal 0.36. And then all you would do is, because you have the Q squared, you want to find Q, so you take the square root of both sides, which then would leave you with Q equals 0.6. So now you have the Q. So can you find the P? Absolutely. You can then go P plus Q equals 1, where P is unknown, but Q is 0.6. And so minus 0.6, and the other side you would put minus 0.6, which would give you a P of 0.4. So that would give you the A allele. Now this is 0.4, so the frequency would be 40%. Now it says, what is the frequency of AA and A little a? This is where you could just put it in, right? So for AA, that's P squared. And A little a is 2PQ. So then the only thing that you're doing here is putting that information in. So you could go, we know that P is 0.4, so that would be 0.4 squared, which equals 0.16, or 16%. This would equal 2 times what P is, 0.4, times what Q is, which is 0.6, which is 0.48, or 48%. All you're doing here is plugging numbers in using these two equations. Now, you would do it for this population, and then if you wanted to see if it was in Hardy-Weinburg, you would do the numbers for the next population, then see, do these frequencies change? If they remain the same, you're in Hardy-Weinburg equilibrium. If they change, you're not. So let's look at the lab procedures here and what we're going to do for your lab. So this procedure 1, experimenting with population genetics. In the lab, we are actually studying rabbit genetics, specifically the gene controlling rabbit fur and furless rabbits. This trait was discovered in 1933 in England by W.E. Castle. Furless rabbits are quite rare because they are more susceptible to cold and usually die in the winter, which makes sense, right? They can't keep their temperature stable to allow their proteins to work and function correctly, which breaks down, and then their organs break down and so forth and so on. In this lab, having fur is dominant, which is a big F, and being furless is recessive. And so we're going to use this information to fill out the actual lab. So these are our cute little rabbits. And so when we're talking about phenotype, we're talking about what they look like. So these two have big Fs, which means they are going to be both furry. This one has a little f, so it's going to be non-furry or furless. Okay? Generally, this activity is done as a class. Each student would mate with another one in the classroom. Bunnies usually have a lot of offspring, but in this experiment, to keep it simple, each couple would have 2 offspring with each mating. The uppercase F card represents the allele for fur. The lowercase f represents the allele for no fur. The lab room would represent the English countryside where the rabbits randomly mate. Each student should head to the front of the room and randomly select 2 cards. These cards represent your genotype and the alleles that you would pass on to your offspring. But today we're not going to do that because we are not in the classroom. But this first generation would represent the initial generation. As a group, you would end up counting up all your FFs, your big homozygous dominant, your heterozygous, and your homozygous recessive individuals in the classroom, and you would enter the information on the table. And so essentially we would calculate it all, and we would end up calculating everything we would need, and we would look at the genotypes, the first, the second, and so on. So what I'm going to do here for you is I'm going to tell you what our total population or what they would look like. And so initially we would have 12 big Fs. We would have 10 heterozygous, and then we would have 8 homozygous recessive. So if we add this all together, we would have 30 in this population. And so from there, we would calculate how many large dominant F alleles we have. Well, if you look here, we have 2 F alleles in these individuals, and there's 12 of those. So it'd be 12 times 2 alleles in here. But that's not the only one we have. We have 1 in here, so we would add 10 to it. So that would be 24 plus 10, which would equal 34 alleles, big ones, right? Now, if we're looking at the lowercase, we would see that we have 8 here. There's 2. So this would be 8 times 2, which equals 16. And then again, we have 1 here, which is 10. So we'd have 26 here. Now, if we add 34 and 26, we would have 60. And you could check this out because we have 30 individuals and each have 2 alleles, so it should equal 60. And then what we would do is how you calculate the frequency of F is you would say you have big F. Well, that's equivalent to P, right? So you do 34 divided by 60 here, which would give you 0.57. And then the other one, you do 26 divided by 60, which would end up giving you 0.43. Now, this is our initial population. So then we would mate. Right? And this is what we'd have. And then I'd say go ahead and mate together, and you know, there's no genetic drift, we have nothing changing. These are the numbers that you would have after your first, your second population, your third, and then all of a sudden, boom, do you notice that 4 through 7 are different? And that is because at this point, it says Siberia because, oh no, at that 4th generation, the environment abruptly changes. Global climate change has caused the temperature to significantly drop in the region. Now, the homozygous recessive bunnies, which were born furless, are killed before they reach reproductive age, so they can't pass them on. They freeze to death. They have no fur. It's cold. And so all of a sudden, if you get a homozygous recessive bunny, it's dead. It died. So instead, you must go to a mating couple and ask them to mate an extra time if that happens. And so then the numbers that you're going to see as you go through, right, there's nothing. What's happening is you're not producing. So these individuals over here, you're going to see more of them because these are dying, right? And then as you go, those are the individuals who are going to be essentially mating with each other. So this is what you're going to see. So essentially what you're going to do here, everyone, is you're going to fill in the number of dominant alleles, the total number of alleles, the frequency of the P and the frequency of the Q, and then you're going to answer some questions. You're going to answer, let me go back here. For instance, you have questions to answer that are going to be for generations 1 through 3. Which basically asks, do the frequency of P and Q change significantly during the time? Is this population currently in Hardy-Weinburg? Is this population evolving? So you need to do that by looking at the calculations that you get from the dominant and recessive alleles. If those alleles remain the same, then the population is not evolving, right? It's under Hardy-Weinburg. If those P's and Q's change, you're in Hardy-Weinburg. And so you're going to look at that from the initial to 3, then generations 4 through 7. And basically, then what you're going to do is you are going to populate in a graph. You're going to put on the bottom, you see generations. So you're going to have at the beginning your initial, where it starts all the way at the x-axis. Then you're going to go generation 1, 2, 3, 4, 5, 6 across. So what am I saying here? So you have this, the initial generations here, and then you go 1, 2, 3, 4, 5. And then they're going to ask you to put the frequency of P and Q. So if this is your first generation, maybe P, one of them is here, and then you can do a different color for the other one, and maybe that one's up here. And then you're just going to follow those as you go through. And then they're just going to ask you other questions afterwards. For instance, the second procedure, looking at population genetics, it says in most populations, humans, we're not able to automatically know who is homozygous dominant and who is heterozygous. Both have the dominant phenotypes. So Hardy-Weinburg equation can be used to calculate the frequency of alleles dominant or recessive and genotypes. Homozygous dominant, homozygous, heterozygous, or homozygous recessive. For example, say in a given population, 49% of the individuals have the recessive phenotype. You have been asked to determine what percentage of the population are carriers or heterozygous. A carrier is someone who has the recessive phenotype. It won't be shown because they have the dominant, but they can give it to someone. You can assume the population's in Hardy-Weinburg equilibrium for the trait. And so you're going to basically take that 0.49% and ask the questions. And they're going to continue on with that question, asking about the entire population. See if you can calculate the changes that are happening in the P's and Q's. And then finally, they're going to ask you some post-lab questions and how you feel, and some more examples of if you can answer the questions or not, and then to reflect on all of this. So I am finished with this today. Hopefully this helped you with Hardy-Weinburg. I appreciate your patience. And if you have any questions, please let me know. I hope you enjoy this lab. I really like Hardy-Weinburg problems. I think they're kind of fun, and you can always work backwards and check yourself. There are tons of internet sources to help you with this. There are lots of videos, so go ahead and check them out as well. Have a great day, and let me know if you need anything.